So far you have seen which are the things: reproduction, vegetative reproduction, sexual reproduction, structure of anther, structure of ovule, then pollination. So far you have seen all these things. You just refer all that, you prepare. I'll just give you a test after completing this lesson. Of course, within 2 or 3 days I will complete you this lesson. Then after that you get ready for one test. Okay. Now, fertilization. So, which is a prerequisite for fertilization? Already you have seen the advantages of pollination. So, pollination, cross pollination, there you have studied about that. Pollination is a prerequisite for fertilization. What is fertilization? The fusion of male and female gametes. Same thing you get in zoology also. Fusion of male and female gametes is called fertilization. Now, in angiosperms, fertilization is double fertilization. See, normally gymnosperms, you get gymnosperms. What kind of fertilization? You get simple fertilization here. In gymnosperm, male gamete fertilizes the egg present in the ovule. So that's what simple fertilization. But here double fertilization. You will see about double fertilization later. Now, double fertilization, events of fertilization. What are the events of fertilization? The first one, the pollen grain deposited onto the stigma. The pollen grain get deposited onto the stigma. What it is? That's what called pollination. Pollen grain get deposited onto the stigma. So that's what germination of pollen on the stigma. So first one, deposition of pollen, that's what pollination. After that, germination of pollen in the stigma. Growth of the pollen in the style. Growth of the pollen in the style towards the micropyle of the ovule. So micropyle of the ovule, in the sense, there's a gap in the integument. This gap is called micropyle. So there only, what happens there? Entry of pollen, growth of pollen in the style towards the micropyle of the ovule. This is what towards the micropyle of the ovule. Entry of pollen into one of the synergies of the embryo sac, that we will see in double fertilization. The discharge of male gametes, then the discharge of male gametes, syngamete, fusion of male and female nuclei, syngamete, and the triple fusion, that we will see in double fertilization. Then pollen pistil interaction. So what happens here? Pollen from one plant get deposited onto the stigma of same kind of plant. Same kind of plant in the sense, for example, potato, tomato, two different, they belong to Solanum tuberosum, Solanum melangina. Potato, Solanum tuberosum, brinjal, Solanum melangina. You just see that here the pollen grain of potato can also be deposited onto the stigma of Solanum tuberosum. So, the pollen grain of potato get deposited onto the stigma of brinjal, but pollination may take place, but the pollen grains will not germinate. So this is what the interaction, that's what compatibility in the sense matching. The pollen grain of one plant can match only for the same plant, not for any other plant. Any other plant is not for any other species. So that's what intraspecific, interspecific. Intraspecific means between the same species. Pollen grain get deposited onto the stigma of same species only. So that's what intraspecific. And then there also you get varieties. There are a lot of varieties. So it may be possible between the varieties also, same varieties, but not between different varieties. So that's what called compatibility. Compatibility in the sense, when pollen grains get deposited onto the stigma of same kind of plant, compatible, matching one. So if it is deposited, or if pollen grain deposited onto the stigma of another different kind of plant, in the sense, incompatibility. So there won't be interaction between the pollen and stigma. The pollen will not germinate. Pollen may get deposited, but the pollen will not germinate. So that's why pollen pistil interaction, the events from pollen deposition on the stigma, pollen on the stigma, pollen. So that's what compatible pollen can germinate. Now in the stigma, there are two different stigmas: wet stigma and pellicle in dry stigma. So wet stigma, pellicle in dry stigma. So in that way, pollination, after pollination, pollen grain germinate. So only in wet condition, pollen grain germinate. In dry condition, the pollen may not germinate. So rejection protein, that's what recognition, rejection protein reaction. So when pollen get deposited onto the stigma, it will germinate. During germination, it will produce certain protein. So that protein interacts with the protein of the stigma. So that time what happens, it favors germination of the pollen, or it may inhibit the germination of pollen. If it is not, even for the same species, there will be self-incompatibility. Pollen get deposited onto the stigma of same plant of the particular species. It won't germinate. That's what self-incompatibility. But only a different plant of the same species, it will germinate. That's what self-incompatibility. So all the factors that will just allow the pollen to germinate or inhibit the pollen germination on the stigma. Then pollen tube in the style. After germination of the pollen in the stigma, I will just cap a block. I will take you in double fertilization, but is given here, but I will take you the block. Pollen tube in the style. After germination of pollen in the stigma, the pollen tube enters into the style from the stigma. So what are the parts of gynoecium? Stigma, style, ovary. Here only the pollen get deposited, pollination, then pollen germinates. So during germination, what happens here? Pollen tube elongates. Pollen tube grows from the stigma, passes through the style and reaches the ovule, through carpel and then ovule. So in that way, the pollen germinates. So that's what given after germination of pollen, the pollen in the stigma, the pollen tube enters into the style from the stigma. There are three types of entry of pollen into the ovule. There are three types of entry of pollen into the ovule. One, porogamy. Porogamy in the sense, when pollen tube enters through the porogamy. The pollen tube enters through the microphyll. That's what called porogamy. When pollen tube enters through the chalasa, it is called chalasogamy. When pollen tube enters through the integument, it is called chalasogamy. So three types of entry of pollen tube into the ovule. And then there are three types of style. There are three types of style. So what are the three types of style? Hollow style or open style. Then solid style or closed style. Third one, semi-solid style or half closed style. Now what happens here? Here hollow style, there is a hollow cavity in the style. There is a hollow cavity in the style which is lined by epithelium. This epithelium secretes mucilage fluid. This mucilage fluid allows the pollen tube to pass through, to grow. So that's what that kind of style is, hollow style or open style. It is common among monocots. The canal in the style is lined by a layer of glandular cells which secrete mucilage, serves nutrition for the growing pollen. Pollen in the sense pollen tube. Solid style. It is common among dicot. Solid style is common among dicot. There is a central core, but of course without any gap. Central core filled with cells. Filled with the cells. What kind of cells? Specialized cells, commonly called transmitting tissue, through which pollen tube grows. Semi-solid style or half closed style. It is intermediate between solid and open type. Then entry of pollen into the ovule. How the pollen enters into the ovule in the sense, but there are three different methods of entry of pollen tube into the ovule. Porogamy. When pollen tube penetrates through the microphyll, it is called porogamy. When pollen tube passes through the chalasa, it is called chalasogamy. When it enters into the ovule through the integument, it is called chalasogamy. So this is all about first about fertilization, then events of fertilization, then pollen on the stigma, pollen tube in the style, then types of style: hollow style, otherwise open style, closed style or solid style. Then second one, semi-solid style or half closed style. So in monocots, hollow style. In dicot, closed style. Now how the pollen tube enters into pollen tube into the ovule? Porogamy, chalasogamy, mesogamy. So what is the microphyll? There is a wide gap in the integument. So this gap is called microphyll. So it will be an opening of seed. When the pollen tube enters through that, that's what called porogamy. When the pollen tube enters into through the chalasa, it is called chalasogamy. So chalasa in the sense, that's a opposite to the microphyll is the chalasa. actually close to the funicle stalk of the ovule. There you have chalasa. Then mesogamy. The ovule is protected by one or two layer covering called integument. When the pollen tube penetrates through the chalasa, it is called chalasogamy. When the pollen tube penetrates through the integument, it is called mesogamy. So this one, orogamy, chalasogamy, mesogamy. Orogamy, chalasogamy. This one is mesogamy. Mesogamy. Then next one about entry of pollen into the embryo sac. So that we will see in the double fertilization. So entry of pollen into the ovule. Entry of pollen into the ovule. Entry of pollen into the embryo sac. What is embryo sac? As I told you already, the ovule surrounding the ovule has a mass of homogeneous cells called archosporia. Archosporium. One of the cell functions as megaspore, mother cell. The megaspore mother cell divides by meiosis, out of which 3 megaspores degenerate, 1 megaspore becomes functional. The functional megaspore gives rise to female gamete of 5. And that's what called embryo sac. This is what called embryo sac. This is the whole ovule. This one is nucellus. Okay. Now you just see that here in that how the pollen tube enters into the ovule. That's the interaction between the pollen tube into the ovule. Now once the pollen tube enters into the ovule through the microfile, what happens? It passes through which one? One of the synergids. Passes through one of the synergids and then reaching into the embryo sac. And then after that, the growth of the one stops. So during that time, what happens here? Male gametophyte, development of male gametophyte or pollen germination. See, pollen has a single nucleus. This nucleus divides into two cells, vegetative cell and generative cell. Vegetative cell remains there, whereas the generative cell divides into two male gametes. Vegetative cell divides to form two male gametes. Sometimes what happens here? Vegetative cell gives rise to tube cell. So that will give rise to pollen tube. So this is controlled by tube nucleus. The growth of the pollen tube is controlled by tube nucleus. Then the generative cell divides into two male gametes. So in the two male gametes reached on, and then now what happens here at the time of germination of pollen, there is growth of the pollen tube. What happens? All the contents move into the tip of the pollen tube. And then this is facilitated by the development of vacuole. Vacuole pushes all the contents into the cytoplasm, tube nucleus, vegetative cell, and then male gametes into that. So that time what happens here? A callose plug is formed between the gametes and the vacuole. Then once at the tip of the pollen tube, there is a hemispherical structure. This hemispherical structure is called cap block. So when the cap block grows, as long as the pollen tube reaches the embryo sac. Once it reaches the embryo sac, what happens here? It won't grow further. That time what happens? Cap block degenerates. When cap block degenerates, the male gametes are released into the embryo sac. So that's what cap block. Now, next one about double fertilization. Double fertilization was first discovered by Navashin and Guinard in Lilium and Fritillaria. Double fertilization is common in angiosperms. Double fertilization is common in angiosperms. It is common only in angiosperms. See, this is what male gametophyte. Pollen. Each pollen produces two male gametes. In the two male gametes, one male gamete fertilizes the egg. One male gamete fertilizes the egg, resulting in the formation of diploid zygote. Another male gamete fertilizes the secondary nucleus to form primary endosperm nucleus. Primary endosperm nucleus gives rise to endosperm. See, the primary endosperm nucleus, triploid one. Actually, secondary nucleus is a diploid one. Then 2n plus n, you get 3n. So endosperm is 3n. Sometimes what happens is it may vary. It may be you may get polyploidy and all that in endosperm. Okay. Now, double fertilization. See, in double fertilization, what happens here? Each male gametophyte, this is what male gametophyte. Male gametophyte. This one is male gametophyte. This is what male gametophyte. So each male gametophyte, in the male gametophyte produces two male gametes. So in the two male gametes, one male gamete haploid fertilizes the egg, resulting in the formation of diploid zygote. Zygote develops into embryo. Another male gamete fertilizes the secondary nucleus to form primary endosperm nucleus, which is a triploid 1, 3N, and then it gives rise to endosperm. So this is what called double fertilization. Why it is called triple fusion? Triple fusion in the sense, here this and all, how this and all formed in the sense. See, this is an obturator. These are not the phenomena taking place during the entry of pollen into the embryo sac. That I will just give you the book you refer from a book. This is what megaspore mother cell. This is what megaspore mother cell. One megaspore mother cell. Megaspore. Mother cell divides by meiosis to form 4 megaspores. Out of 4, already I have studied about the megasporogenesis. There we have studied about it. So 3 megaspores degenerate, 1 megaspore functional, megaspore gives rise to female gametophytes. That's what the embryo sac. Functional megaspore gives rise to embryo sac. That's what female gametophytes. So in that female gametophyte, at first no single nucleus is there. It divides into 2. It divides into 2, and then such 2 nuclei move towards the poles and divide further. 2 into 4, then 4 into 8. 2, 4, 8 nuclei. So in that 8 nuclei, what happens here? One nucleus from each side moves towards the center. 3 form egg apparatus, and the 3 opposite 3 form antipodals. So these two, N plus N, fuse to form secondary endosperm nucleus. Secondary endosperm nucleus. This is what that one. So in that way, what happens here? One first fusion already. Then second fusion, triple fusion. That's why it is called triple fusion. Double fertilization and triple fusion. Okay. Then next we will see post fertilization changes tomorrow. Then after that, what happens here? Certain things given in your book. I will tell you. Then after that, I will just send it to you. The receptacle becomes fleshy and edible around the fruit, enclosing the seeds in Pyrus malus. Pyrus malus in the sense of apple. Then the calyx may persist and enlarge. Solanum melangina may cover the fruit. Physalis minima. Solanum melangina, brinjal. Physalis minima. What is Physalis minima? Physalis minima. Already I told you, the balloon-like, ball-like structure is there. How it is called? Solanacea, which belongs to Solanacea. Refer first to a book. Sodak takali. Physalis minima, Sodak takali. Okay. The next one, the flower stalk axis below the gynoecium enlarges into juicy, pear-shaped body, which is edible. Anacardium occidentale. Actually, the fruit juicy part is not fruit; it's only fleshy stalk of the flower. The perion becomes fleshy in jackfruit. The cells at the tip of the outer interval around the microphile develop into fleshy structure called caruncle, Ricinus communis. Ricinus communis, castor. The funiculus develops into fleshy structure which is often very colorful called aril. Very important, aril. Myristica pithycelobium. Myristica jadikai. Pithycelobium கொடுக்காப்புளின்னு சொல்லுவாங்க, கோக்கலிக்கா கொடுக்காப்புளின்னு சொல்லுவாங்க. ஓகே, pithycelobium. The nucellar tissue is either absorbed completely by the developing embryo sac and embryo, or small portion may remain as a storage tissue. Thus, the remnant of nucellar tissue in the seed is called perisperm. Example, black pepper and beetroot.